One method of genetic recombination in prokaryotes is called transduction. And what I'm going to do in this video is walk through the process of transduction by also paralleling it with the virus life cycle. And so what a virus does, and specifically we're going to be looking at bacteriophages because these are viruses that will only infect bacteria or prokaryotic cells, is infect a host cell. So this image I have here is an E. coli cell for the sake of an example, and we're going to have some kind of bacteriophage with its own gene cargo infect the host cell. And the first thing that viruses do, if it is a lytic virus, is create a bunch of enzymes that will degrade the host genome. And so what will happen after the bacteriophage infects the host is the genome will get chopped up into a bunch of little fragments. And these are going to be caused by restriction endonucleases. And so by this point, the proteins that make up the viral capsid that actually injected the genetic cargo are going to be gone, and the host genome is going to be a complete mess, but there's going to be fragments everywhere. And the viral genome package will exist within the cytosol, and it's going to be active by translating itself into proteins such as DNA polymerase or RNA polymerase to create a bunch more viral capsids and tail sheaths to actually infect other cells. And one of the final things that will happen during the virus life cycle is it will package its genome back into the capsids. So these are capsids for the progeny. And so essentially what happens during transduction is we're going to be getting a lot of copies of the viral genome. But in addition to that, some of these capsids, some of the capsids will obtain fragments of the host's genome. And so this is a host genome fragment. And if the host genome fragment ends up in a bacteriophage capsid, you will have a successful genetic recombination of what could potentially be a beneficial gene. And let's say there was some gene that allowed bacteria to be resistant to penicillin by encoding a beta-lactamase. Well, if that gene stayed intact and was taken up into one of the progeny's viral capsids, and that viral capsid went on to infect another prokaryote, what would happen is this prokaryote will now be resistant to that antibiotic. So you would have its host genome, and for perhaps a short duration of time, if we're looking at a lysogenic virus, that fragment for beta-lactamase, which is antibiotic resistant, will be encoded in another prokaryotic cell, and this would be beneficial because it would be able to survive and reproduce as well, albeit temporarily. And so this is the process called transduction. And so I hope you guys find it useful. Let me know if you have any questions, and thanks for watching.